Assalamu alaikum. My presentation is about basics and applications of fluorescence in situ hybridization, FISH. Introduction to FISH technique. FISH is a common biological technique and a gold standard in diagnostic. FISH is a technique that used for spatial detection and quantification of nucleic acid in their cellular environment. It has emerged as a powerful cytogenic method for the analysis of cells and tissues on the transcriptome and genome level, with more than 6 publications in the literature per day since the late 90s. Principles. The basic element of FISH are DNA probe and a target sequence. Before hybridization, the DNA probe is labeled indirectly via hapten or directly labeled via incorporation of fluorophore. The labeled probe and DNA target are denatured. Combining of denatured probe and target allows annealing of complementary DNA sequence. If the probe has been labeled indirectly, an extra step is required for visualization of non-fluorescent hapten that use enzymatic or immunological detection system. Finally, the signals are evaluated by fluorescence microscopy. Types of probes. Alphoid or centromeric repeat probes are generated from repetitive sequence found in middle of each chromosome. These probes can also be used in combination with locus-specific probes to determine whether an individual is missing genetic material from a particular chromosome. Telomere probes are specific for telomeres and to the 300 kilobase locus at the end of the specific chromosome. Locus-specific probes bind to a particular region of chromosome. This type probe is useful when scientists have isolated a small portion of a gene and want to determine on which chromosome the gene is located, or how many copies of gene exist within particular genome. Whole chromosome probes are actually a collection of smaller probes, each of which bind to different sequences along the length of given chromosome. Procedure. Prepare slides with metaphase chromosome or interface nuclei. Dehydrate in ethanol. Then denature DNA at 70 Celsius. Denatured label probe. Incubate at 37 Celsius for 4 to 16 hours for hybridization. Washing to wash off non-hybridized probe. Detection of fluorescence hybridization signal by fluorescence microscopy. Identification of marker chromosomes. Identify a very small unidentified chromosome or extrastructurally abnormal chromosome. By mixing combination of 5-fluor and using spatial imaging software can distinguish all 23 chromosomes by chromosome-specific colors. This type of analysis can be used to detect abnormalities that affect multiple chromosomes, as in sometimes found in cancer cells or immortalized cell lines. Advantage and limitation of FISH technique. Advantage: high sensitivity, high specificity, rapid, high capacity. Large number of cells can be scored in a short period. Limitations: restricted to abnormalities that can be detected with currently available probes. Cytogenic data can be obtained only for target chromosome. Required fluorescence microscopy and image analysis system. Applications: the identification of specific chromosome abnormalities, the characterization of marker chromosomes, monitoring disease progression, interface FISH for specific abnormalities in case of failed cytogenetics, monitoring the success of bone marrow transplantation. Thanks for listening.